Okay, so we've seen how you make an immune response to a pathogen that's injected into your body. What we're looking at now is using this immune system to make antibodies for antigens that we can use in medicine or for commercial profit. And these are called monoclonal antibodies because they've got one specific use against one specific hormone or antigen. And there are three uses for these monoclonal antibodies. The first one is for pregnancy testing, in which case we make antibodies for the HGC hormone. The second one then is for cancer medication, so for binding and latching onto tumor antigens. And here's how this works. The first thing is you have to get a reluctant volunteer. You get a mouse, and this is a very bad drawing of a mouse, but there you go. There he is. Look how happy he is. Oh wait, his mouth is there. Okay, not biologically accurate. There you go. This is a mouse, and unfortunately, this mouse is gonna be used for science. What we do is we genetically engineer the mouse so that it produces human antibodies, because we can't use mice antibodies because we're not mice. So we've genetically engineered Mr. Mouse to make human antibodies, and now we're going to inject him with whatever antigen we want to make antibodies for. So let's just say we're going to inject him with the HGC hormone, and he's going to have an immune response against that. So Mr. Mouse then is going to have a reaction to whatever it is we've injected in him, be it in this example the HGC hormone, and he's going to start to make antibodies, and these antibodies are going to bind to that antigen. So the immune response happens, and he's making lots and lots of B lymphocytes in there, and that's good because we want those B lymphocytes. The majority of these B lymphocytes are made inside the spleen, and now we need his spleen. And so unfortunately, Mr. Mouse is going to have to be killed. He's going to be terminated, and we're going to remove his spleen. That means that we get to remove also all of his B lymphocytes. All right, so that's it. That's one of my B lymphocytes. Imagine there's many, many more. Now, a B lymphocyte by itself isn't that useful because it won't divide and it won't make more antibodies outside of the mouse. And we want it to divide and make more antibodies. So we bind it to a myeloma. A myeloma is a cancerous tumor cell. We'll bind those two together. That's my myeloma. We're binding that to my B cell, and they're going to produce a new type of cell, and that's a hybrid of the two. So it's called a hybridoma. Now, what's so good about a hybridoma? A hybridoma is a B cell that produces a monoclonal antibody to an antigen that we've chosen, and it divides. So it divides and divides and divides and makes lots and lots and lots of antibodies. We can put this hybridoma into a fermenter or into culture, and we can then extract the antibodies from the hybridoma. So, the run of things, how do they happen? The first thing is we introduce the antigen into Mr. Mouse, so whatever antigen we want, HGC for use in pregnancy testing. So the antigen is injected into Mr. Mouse, who is at this time still alive. This antigen causes an immune response in my mouse. In particular, it's going to cause the primary immune response, and he's going to make lots and lots of B lymphocytes, which I shall shorten down to B lymphocytes, B lymph. Mr. Mouse is then terminated, and his spleen is removed, and so his B lymphocytes are removed, and then those B lymphocytes are fused to cancerous B cells, or myelomas. So, fused to myelomas, and when you fuse them together, you make a hybrid of the two types of cells, which means you make a hybridoma. Now, my hybridoma will divide and divide and divide and make lots and lots of antibodies. You can put that into a culture, be it a petri dish or a fermenter, and then that culture is going to make lots and lots and lots of monoclonal antibodies. And that's it. So you can do that for any antigen that you like, and again, the examples you need to know are uses in pregnancy and the uses in tumor location and destruction or yeah. But that's for another video, which is why we're pausing.